Let me start with a really brief self-introduction, since we're kind of new for conference this time. We are basically a purely mass spectrometry lab. It's a Center for Advanced Research in Biotechnology, Rockville, Maryland. And with a major focus on proteomics of aging. We do all kinds of mass spectrometry, but everything comes into aging eventually. So this short, small project was mostly done by postdoc in lab where we, and it's going to be all about age-related macular degeneration. I don't have time, obviously, in this short talk to go very deep and with long introduction. I really appreciate 2 talks earlier this morning when audience quite well prepared for macular degeneration and retinal pigment epithelium and lysosomal function in these cells and autophagy and about chaperone-mediated autophagy. It's very related topics to my presentation, it helps. So I will go just bullet by bullet very quick with this introduction. So just to say that AMD is a retinal degeneration disease, and probably just to add one typical phrase that it affects majority of population over 65 and a leading case of new blindness in population over 65. It's sort of an extreme picture for our research. And obviously based on sort of common sense and of course based on a lot of experimental data, people believe that senescence and dysfunction of retinal pigment epithelium is a pathogenic pathway for AMD. Key point that these ARPE cells are long-lived post-mitotic cells. It's means they're given us once and forever, and they may accumulate different kind of waste material and to get tired at some moment. So yeah, I mean, it's not a possibility, of course, to do primary cell culture, but we are sort of a very analytical chemistry lab. It's too much for us. We're using just commercially available cells, ARPE-19, which is a non-transformed human RPE cells. Well, it's quite similar to RPE in vivo. Saying quite similar, we always keep in mind that it's just cell lines. It's non-transformed again, but apart from RPE cells in real life, these cells, I mean, it's cell line, it could be supported, it's reaching constraints, it's divided, and it could be split and so on. But that's what we use as a model for our research. And it's based on a recent publication which came from French group and published in Theoretical Biology Medicine recently. What these people did, they treat ARPE-19 cells with this type of peroxide for 1 hour every day and then to let cells to recover for 24 hours and kind of make 5 cycles like this and let cells recover for 3 days and split it, reseed it, and use it as primary cell culture and claim it that that's a model of premature senescent. And this group published like long list of proofs why its cells could be used and qualified as model of premature senescence, which include senescent-associated beta-galactosidase staining and increase in cell surface area, failure to respond to mitogens and cell growth arrest. We reproduced some of this and we treat cells like this, and it's true, that's from my lab. Yeah, control and peroxide treated cells obviously show increased beta-galactosidase staining. They change morphology, they're bigger in size and flatter. So, well, we believe that it's going to be a model of premature senescence caused by recurrent oxidative stress. So, and now it's our turn as mass spec people just to try to analyze and to compare protein profiling between these cells and control cells. Basically, whole idea kind of simple. It's not very much hypothesis-driven. It's more like tabulation. We want to generate long list of up and down regulated proteins or non-regulated, the same level between these two samples. And probably one day it will be published somewhere, but it's not the topic of today's presentation. Today I will present you some data which came unexpectedly, sort of byproduct of this project. But before to jump to real numbers, I really want to show you how we do this mass spectrometry and to convince you that we do reliable measurements, because field of mass spectrometry and proteomics is a little bit kind of overflowed with numbers which hard to reproduce very often. Well, I'm not supposed to make claims like this because I'm also in this field. So two more slides just about mass spectrometry, quick introduction. So just to explain some definitions which I'm going to use, like isotopes, stable isotopes, or exactly second mention on this conference, and monoisotopic peak and isotopic envelope. So the point is that majority of elements have natural abundance of stable isotopes, which are not radioactive, it's just stable. It's proton, deuterium, carbon-12, carbon-13, and so on. And some of them quite a lot. You see it's up to 1% around of carbon-13. I mean, everything everywhere has stable isotopes. So because of this natural abundance and because of very good high resolution of modern mass spectrometers, it means that every peptide on mass spec is represented not by a single peak, but what we call isotopic envelope. It's a bunch, it's a kind of package of peaks separated by one unit. And first peak, it's monoisotopic peak. It means there is no stable isotopes, only normal. You see it says no carbon-13 only. And then it will be one. It could be one carbon or one nitrogen or one carbon, one nitrogen, and so on, whatever. Yes?